Good afternoon, everybody. I think we'll make a start now. My name is Dr. Nick Evans, and welcome to this online lecture. Just a few housekeeping notices before we begin. You can turn on live captions if you want. Please do post questions if you have any in the Q&A. Please note that that is a public Q&A, so do stick to scientific questions. And at the end of the talk, Gail will organise the asking of these questions to me. And the final housekeeping notice is to just make it clear to everyone that this lecture is being recorded and may well be made available later. So moving on to the first proper slide of my presentation, I am delighted to be able to speak about some of the research that I'm carrying out here at Lancaster University, not only because I find it interesting science, but some of the results that you'll see today are in fact results which have been actually gathered by undergraduate students that have worked alongside me in the lab. And so I hope you find the results not only interesting, but also motivating about what can be achieved by undergraduate students as part of their own undergraduate degrees. So I've given a rather long title to this talk. The key words are the final three words: chiral interlocked molecules. And these are a key target of my research program. Some of you, I suspect those of you who are coming to the end of their A-level studies or the equivalent, are familiar with the term chiral or chirality. If you're not, don't worry, I will explain it at the beginning of the talk. But people might be less familiar with the concept of interlocked molecules. And so I intend to actually begin this presentation by showing you what I mean by interlocked molecules. So here are pictures of two interlocked molecules that I actually prepared in my own PhD some 10 years ago now. And these pictures represent the two major classes of interlocked molecule. On the left, we have a molecule which consists of a blue molecular fragment passing through a red molecular ring, or macrocycle as we tend to call it. And this is known as a rotaxane. The red ring is trapped on what is sometimes known as the axial component by the incorporation of large, bulky stopper groups. On the right, we have two molecular rings which are interlocked with one another. And this type of interlocked molecule is known as a catenane. So in these structures, the red and blue components are not covalently linked to one another, but they are a single molecular entity. You can't separate the components without breaking one of the covalent bonds that make up, for example, one of the molecular ring components. So having introduced you to what an interlocked molecule is, I just now want to run through what I'm going to cover in the next half an hour or so as part of this presentation. I'm going to discuss for a few slides what is meant by the term chirality to make sure that everyone knows what chirality is. I'm then going to provide a few more slides on interlocked molecules, including how you can make them chiral. And then I'll discuss a completed research project on a mechanically chiral rotaxane molecular shuttle. Don't worry if you don't understand many of those words at the moment. Hopefully by the end of this talk, you will be able to do so. And to conclude the talk, I want to talk about an ongoing project where we're trying to make synthetic analogues of a naturally occurring class of compounds which are known as Lasso Peptides. So let's start with a simple definition for the term chirality. So any object, and that doesn't need to be a chemical object or a molecule, it can be any object in the world around us, any object that is not superimposable upon its mirror image can be described as chiral. And for those who have met this in their studies, I'm sure you have been shown the example of a pair of hands. So here's a photograph of my hands. You can see that they are approximately mirror images of one another. But if you place one of the hands on top of the other hand, they're not superimposable. So my right thumb is here and my left thumb's over there, so they're not superimposable. And hence, your hand is a chiral object because it's not superimposable upon your other hand. Chirality can arise in many ways in chemistry, and some of you may have met more than one way it can arise. But the one which is perhaps common to all A-level and equivalent qualification syllabuses is chirality arising in organic chemistry, where you have a carbon atom that is bonded to 4 different functional groups. And I'm going to depict an example of this on the screen now. I've drawn out a molecule as we typically draw molecules out at university. So you don't label every single atom with a letter. So where I'm placing my red pointer now is a carbon atom, and it's quite common for us not to actually write out the letter. And that carbon atom is bonded to 4 different functional groups. A hydrogen atom, which is pointing away from us, a CH3 group or methyl group that's pointing towards us, and then an amine and carboxylic acid. If you place this next to a mirror, which is represented by this dotted line, this would be the mirror image. And hopefully you can tell from this diagram that while it is the mirror image, it's not superimposable. So while the methyl group is still pointing towards us and the hydrogen atom is pointing away, the carboxylic acid group is now on the left and the amine group is on the right. And so if we were to move this molecule on top of this molecule, they wouldn't be superimposable. So these are the L and D enantiomers of the amino acid alanine. And more generally, we can say that the pair of non-superimposable mirror images may be called enantiomers. So I've depicted the L enantiomer of alanine here and the D enantiomer on the right. As I'm sure those who have met chirality already in their studies will know, enantiomers of a chiral compound are physically identical except for the direction they rotate the plane of plane polarized light. So they have the same melting points, they have the same boiling points, and they have very similar chemical properties as well. So if you would react a molecule of L-alanine with a non-chiral or achiral chemical reagent, it would react identically to the alanine. However, if you place a chiral molecule in a chiral environment or react it with a chiral reagent, the 2 enantiomers of that chiral molecule will react differently. And this is very important for us as humans because we ourselves are chiral objects. We contain many chiral molecules in our bodies. The most obvious are the proteins that make up, for example, enzymes and receptors in our cells, because proteins are made up of long chain peptides, and peptides are simply covalently linked amino acids, including, for example, L-alanine. So your body is full of enzymes and receptors which contain the chiral centers, and so if they're exposed to a molecule that is chiral, they may well interact with one enantiomer of that chiral molecule in one way and in a different way to the other enantiomer. And this is important when we consume pharmaceutical drugs. And I'm going to illustrate this with two examples. The first is the drug naproxen, which is an anti-inflammatory drug. The enantiomer of this enantiomer, the top enantiomer of naproxen, is the useful pharmaceutical agent. However, the other enantiomer, which I've depicted below, where instead of having the methyl group pointing away, it's pointing towards us, this causes liver damage. And as a result, naproxen is only available on prescription as a single enantiomer. So if you were prescribed naproxen, you would be consuming, hopefully, only the top enantiomer. Most notorious case of this phenomenon is thalidomide. So this was a drug that was developed in the 1950s as a sedative, and because of its antiemetic properties, i.e. it suppresses vomiting, it was marketed in around 1959-1960 as a treatment for morning sickness in Europe and Australia. However, within a few months, reports of increased numbers of miscarriages were reported, and a few months later, children were born with deformed arms and legs. Because it transpires while the top enantiomer, as depicted on this slide, is indeed a sedative, the other enantiomer is terogenic to humans. In other words, it causes harm to the unborn child. And so thalidomide had to be swiftly withdrawn from the market, and it cannot be used for the treatment of morning sickness. So hopefully I've illustrated why chirality is important. The pharmaceutical industry since the 1960s has spent billions of pounds developing new reactions that allow for the access of enantiomer chiral molecules so that we can have safe drugs. And indeed, many organic chemists in academia spend a great deal of their time trying to develop new reactions as well. I have colleagues here at Lancaster University who do that here in the chemistry department. But my particular focus of research interest is in interlocked molecules, the molecules I showed you on that first slide at the start of the talk. And so just to make sure that people are clear on what an interlocked molecule actually looks like, I'm going to illustrate examples of this now by using cartoons. So just to remind you, the Rotaxane consists of molecular rings trapped on a stoppered axial component where we have very large, bulky groups on the ends of the axial to stop the rings falling off, while catenanes consist of interlocked molecular rings. So they look a bit like the Olympic rings. And indeed, someone has made an example of what I would call a 5-catenane, where they have interlocked 5 molecular rings together and called it olympadane. You'll notice that I have introduced some additional nomenclature or terminology on this slide. So for a catenane, if you have a number in front of the catenane, that refers to the number of interlocked rings. In the case of a Rotaxane, it's the number of interlocked components. So if n equaled 1 in this case, what I would have drawn would be a free Rotaxane, 2 rings trapped on a single axle. So interlocked molecules are not trivial to make. They're like, we're getting better at making them. and you'll see the synthesis of an actual Rotaxane in a couple of slides' time. So why do people make the effort to make these interlocked molecules? And the primary motivation has historically been for the possibility of so-called molecular motion. So you could imagine that the ring of a catenane might rotate with respect to the other ring, or the ring of a Rotaxane might rotate around the axle like so. And this is known as pirouetting. Alternatively, the macrocycle or ring of the Rotaxane might move up and down the axle like so, so-called shuttling. And sometimes Rotaxanes, which exhibit shuttling, are known as molecular shuttles. This work was deemed, or the people who pioneered this work, Jean-Pierre Sauvage and Fraser Stoddart, were deemed to have contributed such a significant contribution to the field of chemistry that they were two of the three people awarded the 2016 Nobel Prize for Chemistry for their work on interlocked molecules as the precursors of so-called molecular machines. Along with Ben Ferenger, who was the third awardee that year, who developed molecular machines based on non-interlocked molecules. So I've explained to you again what interlocked molecules are and why people tend to make them, which is to exploit the possibility of molecular motion. So I'm now going to move on to the concept of making chiral interlocked molecules. So you can make chiral interlocked molecules by simply including a carbon atom bonded to 4 different functional groups. And indeed, we have done that here at Lancaster. But you can also make interlocked molecules chiral even if you don't have any carbon atoms that are bonded to 4 different functional groups. The chirality arises simply because you have combined 2 molecular fragments which possess direction, and that can create so-called mechanically chiral interlocked molecules. So I'm going to illustrate this first of all by cartoons. It's not necessarily a very easy concept to understand, but we'll start with some cartoons. So in the top half of the screen, we have what is known as a mechanically planar chiral Rotaxane. This arises when the 2 ends of your axial component are different and the macrocyclic ring is said to have direction. And I'll illustrate what I mean by that with a real example on the next slide. If you have a fully interlocked Rotaxane where the macrocycle is trapped on this axle, the mirror image of that Rotaxane will not be superimposable on the original Rotaxane. It is mechanically chiral. You can get a similar effect with catenanes. So here I have 2 directional rings which are interlocked, and that catenane is then not superimposable upon its mirror image. These are enantiomers of what I would describe as a topologically chiral catenane. So I think it was back in about the summer of perhaps summer 2017, summer 2018, I was lucky enough to have some funding to support a Lancaster undergraduate student to do a summer project in my research group. And the task that fell to them was to prepare a mechanically chiral Rotaxane. And so this is the synthesis they undertook. The first thing they had to do was prepare this new macrocycle here on the left, which they did in 5 synthetic steps. And this macrocycle has directionality. And that's because if you look at the bottom half of the macrocycle below these benzene rings, you'll see that on one side, the structure is different to the other. So on this side, we're missing a carbon atom, which is present on this side. So having prepared this macrocycle, they then dissolved it up in a solvent and then added this amide containing component. So this amide containing component can form hydrogen bonds with the macrocycle as illustrated in the structure of the product. So these black dotted lines are hydrogen bonds. And because of this N3 azide group, you can react with an alkyne. So this is carbon-carbon triple bonds, an alkyne, of course. You can react these two components together using a copper catalyzed reaction to generate the interlocked molecule. And when my PhD student took over this project and optimized the reaction, they were able to get an almost 50% yield of the Rotaxane. And of course, they've prepared both enantiomers of the Rotaxane because there's nothing to favor the formation of one enantiomer over the other. And if you look carefully at the structures, you'll see that no carbon atom is in fact bonded to 4 different functional groups. So these are chiral simply through being mechanically chiral. So how do we know we've made the molecule? Well, we carry out some spectral characterization. So we run proton NMR spectra. So hopefully some of you have met proton NMR spectra as part of your studies already. This is quite a complicated spectrum. It took some time to fully assign it. And we also were able to detect the molecule by mass spectrometry. In effect, you detect the molecule in the gas phase in the presence of an ion, and you'll see that the actual spectrum is very close to the predicted simulation spectrum. And so we're pretty confident we have actually prepared our target molecule. We're also able to detect the presence of the 2 enantiomers of the mechanically chiral Rotaxane by using Chiral HPLC, high performance or high pressure liquid chromatography. HPLC is a very common technique both in industry and in academia for the analysis of molecules. In particular, chiral HPLC is often used to detect and measure the amount or to measure the quantities or the ratio of enantiomers you form in a reaction where you generate a chiral molecule. And what this typically involves is you dissolving up a sample of your chiral molecule, here this mechanically chiral Rotaxane, and injecting it through what's known as a chiral column. So physically, the chiral column looks a bit like a magician's magic wand. It has those sort of dimensions, and it contains a chiral stationary phase. So it's a bit like the human body, where the human body is chiral, so is the internal material of this chiral column. And so if you inject a chiral molecule into it, one enantiomer of that chiral molecule will pass through that chiral column more slowly than the other. And so looking at this graph, what we're basically doing is detecting, we're looking for our molecule coming out. And so after about 28 minutes, the first enantiomer departs from the column. And then after about 32 minutes, the second enantiomer departs from the column. And the 2 peaks are about the same size because, of course, we have a racemate. We have an equal quantity of each enantiomer. So I now want to look more closely at the proton NMR spectrum. And if we look down here at about 2.5 ppm, we have 2 peaks which I've labeled G and G apostrophe or G prime. And these represent the two hydrogen atoms bonded to this carbon atom on the axial component. Now, for those of you who have already studied proton NMR, you would perhaps expect that because those two hydrogen atoms are bonded to the same carbon atom, that they should appear as a single peak with integral or area 2 on the NMR spectrum. They shouldn't appear as separate peaks. And indeed, if you measure the proton NMR spectrum of the free axial, where with no macrocycle present, that is what you would observe. However, because you have a macrocycle, and in particular this rotationary directional macrocycle, these two hydrogen atoms are no longer in the same environment, and so will therefore appear as different peaks in the proton NMR spectrum. However, it is very sensitive to the location of the macrocycle on the axial of the rotaxane. The macrocycle needs to be held by those hydrogen bonding events that I showed you on the synthesis slide at the amide of the axial. If the macrocycle is not over the amide, the peaks will no longer be split. And we can do this in two ways. The first was by changing the polarity of our solvent. So the NMRs I've been showing you so far have been measured in deuterated chloroform, which is the typical solvent we use for measuring NMR spectra. However, you can dissolve the rotaxane up in different NMR solvents, and the one I'm going to focus on is deuterated methanol, CD3OD. And you can see, instead of having two peaks, we now have one peak. It's a bit of a complicated multiplet, but it is only a single peak. And that is because in methanol has disrupted those hydrogen bonds between the amide of the axial and the functionality of the macrocycle. So the macrocycle is now moving rapidly up and down the axial. And so the two hydrogen atoms on that carbon G no longer feel the influence of the directionality of the macrocycle, and so appear as a single peak. We can get a similar effect by adding acid and base to the macrocycle, or more specifically acid. So here I have our close-ups, like a snapshot of a section of the proton NMR spectrum recorded under different conditions. So the first spectrum, the one at the bottom, spectrum A, has the two clear peaks representing the two hydrogen atoms G and G prime that we have previously observed dissolving up the rotaxane in deuterated chloroform. If you now add acid, and that's what I'm doing as I move from A to B to C to D, etc., the peaks move and coalesce into a single peak. So spectrum H, we only have a single peak. And that is because, as illustrated by this diagram, this cartoon, what we've now got is, instead of the macrocycle sitting stationary close to the two hydrogen atoms of interest, the macrocycle is now moving up and down the axial of the rotaxane, and so it's spending a good proportion of its time away from the two hydrogen atoms, and so the two hydrogen atoms don't feel its presence, and so they are now equivalent, and so they give a single peak. If you add base, all the protons that you have been adding to various functional groups on the rotaxane will be removed. Acid plus base equals salt plus water. And so the macrocycle can return to being over the amide, forming hydrogen bonds, and we get a return of the two peaks for hydrogen atom G and hydrogen atom G prime. So that's the complete project on this mechanically chiral Rotaxane molecular shuttle. And we published that in 2019, and that was nice for me, my PhD student, and the undergraduate student. But a perfectly valid question to ask at this point is, this is all very nice, but this is very fundamental research, isn't it? Does it actually have any use in the real world? And to conclude this talk, I want to just provide you with some indication of how I think this could be useful in a real world application. So on the slide, I'm depicting the chemical structure of a naturally occurring product known as a Lasso peptide. So as you can see, we have a molecular fragment passing through a molecular ring. And so in effect, we have almost a Rotaxane. The difference from the Rotaxanes we've seen previously is that there is a covalent bond between the acyl-like component and the macrocyclic ring itself. And so if I was describing this structure, I'd actually describe it as a one Rotaxane because there is only one molecular fragment. As far as we know, humans don't form this type of molecule, but a range of microorganisms do. And they've been found to have useful biological properties. And I've listed some here. And the ones which intrigue me personally the most are things such as antimicrobial or antiviral behavior, because it suggests that these sort of molecules might be useful as pharmaceutical drugs. The problem is that they are clearly very complex molecular structures. And indeed, there is very little in the chemical literature, or there are, very few reports in the chemical literature describing the synthesis of these molecules by chemical synthesis. The only examples of Lasso peptides you'll find are ones which are naturally occurring or variants which have been prepared by biochemists using biochemical methods. However, a few chemists have proposed, including ourselves, that what we should do is try to prepare simpler analogues from two Rotaxanes, where we take a standard Rotaxane and then link the macrocycle and acyl components of that Rotaxane with a biologically interesting peptide sequence. And so on the next slide, I'm going to show you the work we've done so far on this. And this work was done in part by an undergraduate student as part of their undergraduate degree here at Lancaster. So the first thing we did was prepare the two Rotaxane, like the standard Rotaxane and macrocycle, trapped on a stoppered acyl. And the synthesis is very similar to the previous synthesis we saw earlier in this talk. Because of the presence of this amine group, you can attach a short peptide chain by an activated coupling reaction to the ring of the macrocycle. And then by removing the chemical functionality in black, which are known as protecting groups, and then doing a second activated coupling, you can cyclize to form a one Rotaxane, where I've included a short, simple peptide sequence between the macrocyclic ring and the rest of the acyl component. So we reported this in 2020, last year, and not only have we reported upon this, this was a key result in a successful grant application for myself to allow me to employ a senior research associate at Lancaster to prepare further examples of these one Rotaxanes, to look to include different peptide sequences to see if we can replicate useful biological properties in these synthetic analogues. And as part of this study, Beck or Rebecca will be looking to incorporate a range of directional macrocycles to see if we can identify a difference in biological behavior depending on whether we have one mechanically chiral isomer compared to the other. So that concludes the science. I just wish to acknowledge a few people because not all the work I presented is my own. So Beck is my current senior research associate who's continuing the Lasso Peptide work. Charlie was my PhD student who helped me finish off the mechanically chiral Rotaxane molecular shuttle, while Tim and Matt were the two undergraduate students who helped with the projects I've described, both of whom have gone on to do PhDs elsewhere. I also wish to thank various agencies for funding my research. And to absolutely conclude, I wish just to point out where you might be able to find some further information. I have my own research website, which you may find online. And I would recommend that you do go and check out the Nobel Prize lectures on molecular machines. They were the Nobel Prizes in Chemistry in 2016, because not only do you find now on the Nobel Prize website the printed text of the Nobel Prize lectures, but the more recent Nobel Prizes, including the 2016 Nobel Prizes, are also available as videos. You can watch the Nobel Prize laureates present their research on a video. So I think that concludes the talk. Thank you very much for listening, and I look forward to any questions anybody may have.